In the previous video, you learned how some proteins can be activated by post-translational proteolytic processing. I gave you two examples, the digestive enzyme chymotrypsin and the hormone insulin. In this video, I'll present a more complex case showing how this type of processing can be responsible for the activation of a full cascade of reactions leading to blood coagulation. Before I describe the formation of a blood clot and the coagulation cascade, I want to start with an analogy. Imagine an old house. Like in many old houses, this house contains a continuous closed pipeline heating system carrying heated water or steam to all rooms in the house. If one of the pipes springs a leak, it would affect the complete heating system, and the leak has to be fixed. To avoid any serious consequences, you have to call a plumber who would bring some material to plug up the hole and restore the integrity of the heating system. Would it be amazing if your pipes contain a self-sealing compound that could stop leaks anywhere along the heating system? You would not need to wait for a plumber, and the consequences of the leak would be minimized because the self-repairing mechanism will be initiated very quickly. The house with the closed circulating system is similar to the human body. In a human body, the cardiovascular system is equivalent to the house heating system. It is a closed system where water or steam are replaced by blood. Unlike what circulates in the heating system, blood contains self-heating molecules that can form blood clots to plug up holes and cuts due to vascular injuries. What are these molecules that make up the blood clot, whose function is to plug the lesion in a blood capillary? In human, blood clots are made of two main components. First, platelets that form a plug to stop the flow of blood out of the damaged vessel. Platelets are cellular fragments devoid of nucleus that derive from bone marrow cells called megakaryocytes. Second, fibrin, a fibrous protein, forms a mesh that extends outside the blood vessel and stabilizes the platelet plug. To understand how the flow of blood exiting a blood vessel's lesion is quickly repaired, we need to answer two important questions. The first question is the following: how do platelets and fibrin work together to carry out blood clotting, also known as blood coagulation? To answer this question, I will discuss three phases in the process: initiation, extension, and stabilization. The second question that we need to answer is the following: how is the blood clotting function regulated by the coagulation cascade? So let's start with initiation, the first phase of blood clotting. This phase is triggered when the blood comes into contact with a breach or cut in the wall of a blood vessel. The lesion exposes collagen fibers that are normally found in the extracellular matrix of the tissue surrounding blood vessels. An inactive form of the von Willebrand factor is always circulating in blood vessels. When it comes into contact with collagen, this factor attaches to the collagen fibers. Shear forces due to the circulating blood result in the activation and unfolding of the von Willebrand factor. This unfolding exposes binding sites on the von Willebrand factor that can then attach on the surface of platelets. So platelets start to attach to the lesion sites and to accumulate. Attached platelets get activated. The activation promotes the expression of adhesion molecules at the surface of platelets. The result is a recruitment of additional platelets. Extension, the second phase of the blood clotting process, consists in the recruitment of additional platelets and the formation of a more stable aggregate. The increased cohesion between platelets at the site of lesion is in part triggered by the interaction between integrins, adhesion proteins located at the surface of platelets, and a circulating protein called fibrinogen. Each fibrinogen molecule has two binding sites for integrins. Therefore, platelet-platelet interaction is made possible when each binding site on fibrinogen interacts with adjacent platelets. The release of ADP and thromboxane A2 by activated platelets attached to the site of lesion contributes further to the adhesion and aggregation of platelets. The outcome is the formation of a soft plug at the site of lesion. During the last phase, stabilization of the clot is the result of the conversion of fibrinogen into fibrin and the polymerization of fibrin into a network of fibers. These fibers stably anchor the platelet plug at the site of the lesion. The conversion of fibrinogen is catalyzed by a serine protease called thrombin. In summary, the activation of platelets occurring during the three phases is driven by four factors. Collagen, exposed when a lesion occurs, is the first factor that promotes the recruitment and activation of platelets. The second factor is thrombin, that initiates the coagulation cascade by triggering the formation of a fibrin cloud. The role of thrombin will be discussed in more detail later in the video. The last two factors, ADP and thromboxane A2, are agonists, that is, molecules that initiate physiological responses. They are produced by activated platelets and contribute to recruit and activate more platelets. Thrombin, ADP, and thromboxane A2 diffuse from the base of the platelet aggregate outward. The result is a formation of a clot with a core made of fully activated platelets and a shell of less activated platelets. Another consequence of this gradient of factors is that the platelets in the inner core are more closely packed and more stable due to a high level of interplatelets interactions and to fibrin deposition at the base of the clot. In contrast, platelets in the outer shell are loosely packed and less stable because fibrin deposition is very limited in this region, and the interplatelets interactions are dependent on the relative concentration of ADP and thromboplaxin A2. Fibrinogen is the third most abundant protein in blood plasma. This 340 kilodalton protein consists of two C-terminal domains, here labeled C, connected via coil-coil regions to a central nodule labeled N on this figure. How does thrombin convert a fibrinogen molecule into a fibrin monomer? Thrombin cleaves a small portion of the central nodule, releasing 18 to 20 amino acid long peptides called fibrinopeptides. The remaining part of fibrinogen becomes the fibrin monomer. The cleavage and release of fibrinopeptides reveal binding sites on the central nodule, here represented by blue diamond-shaped motifs. These binding sites interact with the C-terminal domain of other fibrin molecules. The two binding sites on a given fibrin molecule interact with the C-terminal domains from distinct fibrin molecules. The result is the polymerization of fibrin into a meshwork of fibers. Finally, coagulation factor 13 catalyzes the cross-linking of fibrins to stabilize the fibrin filamentous network. The fibrin meshwork extends from the base of the platelet plug into the extravascular region around the lesion. Without fibrin, the platelet plug would become loose, pushed by the blood flowing in the blood vessel, and the clot would never form. Of course, the fibrin meshwork should form only when needed, in case of lesion. We know that fibrin is not formed until thrombin cleaves fibrinogen. The formation of fibrin is thus controlled by the activation of thrombin. In turn, this activation is regulated by the coagulation cascade, a process triggered by the formation of a vascular lesion. Let's look in more detail at how a vascular cut can activate the coagulation cascade and ultimately the thrombin-mediated fibrin polymerization. As you can see on this cross-section of a blood vessel, the layer of cells lining the lumen of the vessel is called the endothelium. Surrounding the endothelium, a second layer of cells is characterized by the expression of a transmembrane receptor called a tissue factor, or TF. As you can see on the side view of a blood vessel, a vascular lesion damaged the endothelium and exposed the tissue factor presenting cells to the blood flowing in the lumen of the vessel. The coagulation cascade is divided into two pathways, the extrinsic and intrinsic pathways. The extrinsic pathway is activated first and provides an initial burst of thrombin production and formation of fibrin filaments. The intrinsic pathway activation is initiated by activated platelets and plays a role in the sustained production of fibrin and in the clot maturation. Let's look first at the extrinsic pathway. At the site of lesion, the exposed tissue factor acts as an intrinsic initiator of the clotting process. The tissue factor is now exposed to proteins circulating in the blood, in particular the inactive form of the coagulation factor 7. Note that by nomenclature, all the coagulation factors are identified by Roman numerals. The interaction between TF and coagulation factor 7 leads to the activation of the coagulation factor. Another point of nomenclature, the subscript A placed after the Roman numerals indicates that the coagulation factor is in its active form. The formation of the TF coagulation factor 7 complex increases the serine protease activity of factor 7A by several orders of magnitude. Active factor 7A cleaves coagulation factor 9, which becomes factor 9A, another activated protease. Activated factor 9A requires activated factor 8A to convert the inactive serine protease coagulation factor 10 into its active form 10A. Then factor 10A binds to the complex formed by the tissue factor and factor 7A. Another activated coagulation factor, factor 5A, is recruited by the complex. Together, factor 10A and 5A cleave the inactive prothrombin into thrombin. Thrombin is the active form of the protease capable of converting fibrinogen into fibrin and in activating platelets. This pathway can be initially activated because there are small amounts of circulating factors 8A and 5A. However, the bulk of these two coagulation factors is formed by thrombin-mediated cleavage of their inactive forms. This is an example of positive feedback. Thrombin is also responsible for the cleavage and activation of factor 13. As you recall, factor 13A is required for the cross-linking and stabilization of the fibrin meshwork. What about the intrinsic pathway? Studies suggest that several factors, including misfolded proteins, bacterial surface proteins, or exposure to anionic compounds such as extracellular nucleic acids or phospholipids displayed at the surface of activated platelets at the site of the vascular lesion, initiate the intrinsic pathway. Exposure to these factors triggers the activation of the coagulation factor 12. Factor 12A is once again an active protease. Factor 12A cleaves factor 11 to produce factor 11A, an active protease. From that point on, the intrinsic and extrinsic pathways share common steps. Factor 11A that has just been activated is responsible in conjunction with factor 8A for the activation of factor 10. Both pathways contribute to the processing and activation of thrombin. The outcome is an enhancement of the formation of the fibrin's meshwork and the activation of platelets. While the intrinsic pathway does not appear to have a major role in the blood clotting process, when misregulated, this pathway might contribute to thrombosis, that is, the formation of blood clots in the absence of vascular lesion. These blood clots can block the blood flow in blood vessels, leading to serious health issues. Inhibiting the intrinsic pathway appears to protect against thrombosis without significant risk of bleeding uncontrollably. Now that the vascular lesion has been sealed and plugged up, how is the coagulation cascade stopped? Ironically, thrombin plays a central role in the deactivation of the coagulation cascade and in controlling the size of the blood clot. Over time, thrombin forms a complex with protein S. Then thrombin and protein S form a ternary complex with thrombomodulin, a membrane protein found at the surface of vascular endothelium cells on the lumen side of the blood vessel. The ternary complex triggers the activation of protein C, a protease that degrades factor 5A and 8A. The result is the inactivation of factor 9 and 10, and ultimately the coagulation cascade comes to a stop. There are two additional levels of inhibition. First, protein TFPI binds and inhibits the tissue factor as well as factors 7 and 10. Second, the protein antithrombin 3 binds irreversibly to thrombin, inactivating its proteolytic activity. Antithrombin is also responsible for the inactivation of other serine proteases, including coagulation factors 9, 10, 11, and 12. Therefore, both the intrinsic and extrinsic pathways are blocked. In summary, I discussed the three steps of the blood clotting process: initiation, extension, and stabilization, as well as the regulation of the coagulation cascade. The coagulation cascade is how the body carries out hemostasis. Hemostasis is the medical term for stopping of the blood flow. The coagulation cascade is an important response to vascular lesion because it maintains the vascular system as a closed system. As such, hemostasis must be tightly regulated. Too little activation of hemostasis leads to significant and dangerous blood loss. In contrast, too much and unregulated activation of the coagulation cascade can lead to thrombosis and the formation of unwanted and dangerous blood clots. In another animation, you will learn how radiologists can monitor the medical effects of thrombosis.